Okay, good morning. Let's continue our discussion about the cell cycle. And we will talk today about how it is that the cell actually moves from one stage of the cell cycle into the next. What are the different types of factors that are controlling that? How the cell actually manages to interpret different signals from its surroundings that tell it to go ahead and move through the cell cycle, and what happens inside of the cell involving many different proteins that control this progression. And at the end, we'll also talk about a very special phenomenon of living cells, especially cells in our body. That is the ability of cells to undergo apoptosis, which is programmed cell death under certain circumstances. We last time discussed that the different stages of the cell cycle, right, can conveniently be thought about as sort of like a clock, right, or a full 24-hour cycle, even though I emphasized that notion of somehow timing it isn't really practical, right? Because it's not so much about how much time has elapsed for a cell to be triggered to move into the next stage of a cell cycle. It has more to do with different conditions, different types of signals that the cell is receiving that tell it to progress, and also the different stages are all stages where certain things have to happen and happen properly. If things don't happen properly during a stage, then it might take longer for the cell to move on to the next stage. So just to review, when we say cell cycle, what we're talking about are all of the activities that a cell undergoes between cell divisions, right? From the time that a parent cell divides into two daughter cells, right? You can think of that as the start of the cell cycle for each of those daughter cells. And eventually, maybe those daughter cells just continue doing their normal cellular functions like they are programmed to do, depending upon what type of cell they are. But maybe at a certain point, they are told, oh, you should go ahead and divide. Right? So at that point, they would then continue on through their own cell cycles, and then each of them would divide into two new daughter cells as well. So really, again, it's not so much about a timed kind of thing, like you have a stopwatch and now you're trying to time how the different stages work, or the cell somehow uses a timer like that. But it's a series of processes and decisions that the cell has to make, depending upon signals that are coming from the outside, as well as signals that are coming from within the cell itself. So the timing then really varies greatly, depending upon the cell function and the conditions. For example, in this cartoon here, this is a nice reminder that the four main parts of the cell cycle that we're concerned with are the G1 phase, which is where the cell, if it's a fully differentiated cell in your body that's carrying out its fully differentiated cell functions, whether it's a nerve cell, a muscle cell, skin cell, hair follicle cell, or a gland cell somewhere, right? During G1 is when it's doing those normal functions. And so a fully differentiated cell would probably spend most of its time just in G1, happily doing its job, right? If it is told that it should go ahead and start dividing, then it will put on hold all of its normal G1 functions, right, and switch over to the task of replicating its genome. And so that would be S phase, right? And we said that it roughly takes about 8 hours for a cell in your body to be able to replicate or copy its entire genome, right? All 3.2 billion base pairs that make up your 22 pairs of autosomes and pair of sex chromosomes, right? Then once the DNA is copied, the cell goes into the G2 phase. This is where, it basically evaluates whether or not the copy of the genome was successful, or if there are any real problems that need to be addressed with it. It's also when the cell will start making or transcribing a particular set of genes to make the protein products from these genes that are going to be necessary for mitosis. And if all that goes well, Then you enter mitosis. And as we looked at last time, mitosis is that roughly 1 hour process where the chromosomes fully condense into those really tight, stubby rods, right, that we're used to seeing, right? And at that point, there will be these X's because each one will be a chromosome that has duplicated itself, right? And those two sister chromatids, the original chromosome and the copy, are still stuck together, right? So they make this sort of X shape, right? And then those two sister chromatids are going to be pulled apart to either side of the dividing cell so that each of the daughter cells then gets a full copy of the genome, right? So that process of mitosis we looked at is in 5 steps: prophase, prometaphase, metaphase, anaphase, and telophase. Right? So depending upon what type of cell you're talking about in your body, some cells go through this pretty quick and pretty frequently. Others, might not go through this cell cycle very much at all. They'll just kind of stay in G1 doing their job until they die, right? And some of them, in fact, don't even divide at all. So some examples: cells that divide very rapidly are the cells, for example, that are the precursor cells that make your red blood cells. So your red blood cells are erythrocytes, right? So the cells that are differentiating and becoming those erythrocytes, the very last stage are the erythroblasts, right? And then they differentiate further and divide and become your red blood cells. Right? Slow cycle cells are things like, liver cells, certain types of immune cells, certain glands. They divide less frequently. And then some cells don't really divide at all. So I think you're probably aware, right? Like your nerves, your neurons, they don't divide, right? They don't reproduce at all. And that's why if you have sustained certain types of injuries to your nerves, like spinal cord injury, right? You cannot just regrow or recover, right, by replenishing your spinal cord nerve cells, right? So that's a big problem. So the other part of this, which I don't remember if I mentioned last time, but I will mention it here, is this G0. Right? G0 is an arrest. So in other words, if you look at the way the arrow is drawn here, it's almost like the cell is taking a break from the cell cycle and going into G0. When I used to live in California, and when I was doing my undergraduate time up in the San Francisco Bay Area at UC Berkeley, my home where my family was, my mom and dad, they were back down in Los Angeles. And so I got very used to sort of driving up and down during breaks from school, right? Whether it's like spring break, Thanksgiving, even a long weekend, sometimes I would just drive down. And that's like a 6-hour drive, right? California is pretty stretched out. So I'd be driving along the I-5 highway or the freeway most of the time, and it's super boring, right? So every so often, if I came upon a rest stop, if I was getting really tired and not feeling like I should keep driving, I would pull off into the rest stop, maybe get out of the car, walk around a little bit, take a nap, whatever, until I felt good again, right? And then I'd get back on the freeway and resume my journey. Right? So G0 is like that in cells also. Remember, the cycle that's drawn here, it's not really like the cell was traveling somewhere, right? These are all changes that are happening inside of the cell, representing different phases of the cell cycle. So if certain conditions aren't quite right, and the cell isn't so happy with these conditions, I mean, it's not just going to die, right? But it doesn't want to continue doing its normal functions because maybe there's a lack of resources, nutrients, whatever. It may go into G0. That's like pulling off the freeway, taking a rest until things get better, and then going back on again. Right? That actually is very different than the other type of cell arrest, which we call senescence. Right? We don't really talk about senescence that much in this class, but it's a very important concept when we talk about things like cancer, for example. Right? Cells that go into senescence cannot resume the cell cycle. So why is it that most of the time fully differentiated cells can be thought of as just hanging out in G1 and being very content, not really moving on in the cell cycle? Think about this, right? Our cells are parts of tissues, right? So a tissue in our body, right, is an environment made up of many, many types of cells, but you might have an abundance of a particular type, right? And each one of these cells within the tissue is doing its job, right, to contribute to that type of tissue. So think about the tissue as a busy office with lots of cubicles and lots of employees. So these employees, most of them at any given time, right, are doing the specific jobs that they were hired to do, right? That's why they're there. Right? So that's like the individual cell in the tissue that it belongs in. It's there, this fully differentiated version of this cell, to do its job. Right? And they're just focused on doing their job and doing it properly. And that's why the cells will stay in G1, right, until they're told to do something otherwise. So if the tissue is healthy and happy, there's enough of the tissue, and it's functioning properly, there might not be any reason for many of these cells to worry about dividing or changing. So when you do have a cell that gets the message to divide, it's sort of like if you have a new employee joining this office, right? And the new employee, just like you, when you've taken a new job or something like that, right? You have to spend maybe part of your time at first, right, just doing stuff like HR paperwork, right? Administrative stuff to get you set up, get on payroll. Maybe you get training, something like that, right? So that part of the new job is very important, but that's not what you were hired for, right? You weren't hired to attend an HR training orientation, or you weren't hired to actually, fill out tax form paperwork, right? You were hired to do a particular job. So if you think about it, a cell that has been told to divide is like undergoing this process where it takes a break from doing the job it was hired to do, and it's now kind of getting set up again, right? Like setting up your cubicle when your new boss tells you, hey, this is your seat over here. Now, for stem cells, it's a different story, because stem cells, their whole job is to keep multiplying, right? Remember, I said cells in our body are either going to be doing the job that they're meant to do, or they're worried about dividing, right? And they cannot do both at the same time. Stem cells, of course, are sort of an exception, because in the case of a stem cell, its job is to divide and replenish all the other differentiated cells in our tissues that are being sort of turned over, right? Worn out, damaged, recycled, whatever, right? So as a cell is going through these different stages of the cell cycle, it would make sense that because each stage of the cell cycle is a critical set of processes that must be done properly in order for the cell to arrive at a point where it's ready to actually split in mitosis, you have to imagine that there are certain checkpoints along the way where the cell can assess whether everything that it was meant to do in that particular cycle was successfully done and there are no problems or mistakes that might be, if they're allowed to be carried on, right, lead to a really bad situation. Mainly, the mistakes we're talking about are mistakes in the DNA replication. Certainly by now in the semester, all of you have an appreciation for this concept of each nucleus in each of our nucleated cells, right, carrying the exact same copy of the genome that makes us, right? Or you should say two copies, one from mom and one from dad, right? And together, that is the genome that makes you. So in order for a cell to successfully divide and still function properly, right, you better make sure that the process of duplicating the genome, right, and the process of properly allocating or dividing it up evenly between the two daughter cells, right, is done with great care and great precision. Right? Because if you give a daughter cell a wrong complement or a wrong allotment of genes, right? Or if the genes that you're giving them are damaged because during chromosomal replication, the chromosomes got messed up, right? That can lead to all kinds of trouble, right? Maybe in a best case scenario, two daughter cells that are messed up, right? Maybe they just die because they can't function properly. And then it's kind of like, well, whatever, right? These are just two cells out of the trillions of cells in your body, if they just go away because they're messed up, no harm, no foul, right? But what if the mistakes that are involved in duplicating the genome and dividing them up to 2 new cells results in very strange, messed up versions of these cells, right, that don't function properly and might continue dividing, right? That can lead to diseases like cancer. Right? So when you think about at any given time, how many millions of cells in your body today will be dividing as stem cells, right? And how many millions upon millions of cells, differentiated cells, might be dividing just to kind of replenish, some more of the tissue. It's kind of scary, right? To think that each one of those hundreds upon hundreds of millions of cells that are dividing today have to get it right. Okay. So there are checkpoints along the way. So the main checkpoints are between the G1 and the S phase, which we call the restriction point. And then there's an important one between G2 and mitosis, and then one towards the end of mitosis, the metaphase-anaphase transition. So these, just like the word checkpoint suggests, are opportunities for the cell to make sure that everything went right. Okay. So the restriction point in particular is of interest because remember I said most differentiated cells will be just completely content hanging out in G1 doing their job, right? So if they receive signals that say, no, take a break from your job, right? You need to go ahead and do this now, like head towards dividing by replicating your genome first. Right? You better make sure that you're prepared to undergo or enter S phase, right? So the restriction point is that critical checkpoint. You can think about it this way. When I was a kid, I kind of had this like love-hate relationship with roller coaster rides, right? On the one hand, I loved roller coasters because they're thrilling. On the other hand, I was scared to death by them, but that's why they're thrilling, right? And so I remember I'd often go with my friends to amusement parks, and as we're sort of waiting in line, right, I'm sort of like, talking to their friends, we're all goofing around and stuff like that, but it's almost like I'm building up courage to get ready to get on this ride, right? And a lot of times you'd be in this line, and right at the end of the line, before you get on the ride, there was the little gate. We called it the chicken gate, right? Because that's like your last opportunity to kind of bail on the ride, right? You can get out of line. But of course, none of us would do that because then the rest of us would make fun of him or her, right? But during that time, right, you still have the opportunity to kind of bail, right, on actually getting on the ride. But once you pass the restriction point, meaning, hey, once you get in that roller coaster car and you're strapped in, that's it, right? You are committed to seeing this through to the end of the ride, right? As the roller coaster is going up to the top of the first hill, you can't be like, nope, forget it, right? You can't just like take your strap off and like, get off, right? You are stuck. So the cells in our body are like that as well. Once you pass a restriction point, right? Once you pass the restriction point between G1 and S phase, all right, the cell is committed to seeing this all the way through to mitosis, right? It cannot back up and decide to go back into G1. Right? So what makes this process a commitment, right? What's actually happening inside of it? Well, one thing that we've been talking about a lot this semester is that the critical functions of cells inside are controlled by proteins, right? Proteins that are encoded for by your genes, proteins that are specific three-dimensional shapes that serve as little machines. We've certainly looked at many examples of proteins that interact with other proteins because they're the right shape, and that's how they do different functions, right? So hopefully it's not surprising to you that the building up of different changes inside of the cell that lead it to a checkpoint like the restriction point and ultimately push it over that checkpoint have to do with the buildup of certain proteins inside of it, getting ready to carry out particular functions, right? For example, if you're in G1, right, maybe there are certain proteins that you normally don't care about making in G1 because they're not part of you being a good nerve cell or muscle cell or gland cell. But when it's time to replicate your genome, hey, where are those proteins, right? Where are things like your DNA polymerase, right? Where are things like those various enzymes that will help make those replicon bubbles and start actually making Okazaki fragments, right? You're not going to have a lot of those in G1, because if you're just going to be doing stuff in G1, you don't care about having those types of proteins around. But if you're told, hey, no, you better start going through the rest of the cell cycle, you have to start building up levels of those proteins. And once those proteins reach a certain amount, it's almost like you're ready now, right? And that wave of special proteins that are meant to be used in the next phase, in S phase, is what's pushing the cell over the restriction point. Early experiments really brought attention or focus to this concept of it's the buildup of different proteins. For example, look at these experiments. These are kind of cool. If you take two different cells of the same type, right? Let's say you have a bunch of cells that are growing in a culture, just like those pictures of the onion cells that we looked at. You can find some that are in mitosis in different stages, right? You can find some that are maybe in S phase, right? Most of them would just be in G1. So what early researchers did was they took two of these cells, one that's in G1, just minding its own business, doing its normal differentiated function, and one cell that was already in S phase. And what they did was they fused these two cells together, right? So you get this weird cell that has two nuclei, but importantly, what? All of the proteins that are contained in the cytoplasm of these two cells are now mixed, right? And so what they found is by doing that, the other nucleus, the green one that used to come from the regular G1 cell, right? It also begins to change, right? And it starts showing the signs of it's going into S phase now, right? So what does that suggest? That suggests the buildup of certain proteins that pushed this cell, the one on the left, into S phase, right? Now that you're actually exposing all those proteins, right, to the nucleus in the G1 cell, right, it also makes that cell start going into S phase. So you can do this even more dramatically. Check this out. Take another hapless G1 cell that was minding its own business, doing its normal functions, and you find another one of those cells that's right in the middle of mitosis. All right, so look, the chromosomes have already condensed into these rods. The nuclear envelope is beginning to dissolve, right? And it's going to start coming apart as two daughter cells. What if you fuse these two together? Well, what's really scary is the same principle applies, right? This cell that was undergoing mitosis is loaded up with whatever proteins were made during G2 that made it ready to be able to do mitosis, right? Now, all of a sudden, those proteins are being sort of dumped into this poor hapless G1 cell on the right, and that G1 cell starts doing mitosis, right? But that's really freaky. Why? Because it's completely skipped S phase, right? That G1 cell never had a chance to duplicate its genome. So as a result, whatever single copy of the genome it had, that begins to condense into rods, right? So you can see, rather than getting the normal proper example where you have two of these chromosomes, one is the original, one was the one that was copied during S phase, and now they're condensed, getting ready to be pulled apart, right? Whatever the G1 cell had, it's just forced to go ahead and go into this process as well, right? Crazy experiment, right? Obviously a very unnatural situation, because you would not have two cells that would spontaneously fuse like this. And that's a good thing, right? We take it for granted that that doesn't happen because our tissues in our body, right, would never be able to function if you had cells spontaneously being forced to undergo mitosis by being fused with some other cell that is properly undergoing mitosis. Right? So we call these experimentally fused cells heterokaryons. Remember, karyotype is a name for that image of all the chromosomes that are lined up, right? That's your karyotype. So a heterokaryon cell is one that has mixed genomes together, right? The genomes of two different cells that are now combined together into one. Okay. So hopefully you appreciate that what's happening in the background as cells are moving through the cell cycle from G1 to S, G2, and into mitosis is the buildup of different protein levels that are needed for the next stage to kick off. Right? So we can look at this using this figure here. This is a nice figure because I think it highlights an important concept. So on the x-axis is time, maybe over many hours or many days. On the y-axis is the relative concentration or amount of a particular protein that you're interested in studying inside the cell. So early on, researchers saw that there was a particular protein that we'll just call MPF, or mitosis promoting factor, right? And when you have a lot of this protein, then the cell is triggered to go into mitosis. So of course, if you overlay the black line, so the black line is the rising and falling of levels of this MPF protein in the cells, if you kind of match it up with what stage of the cell cycle this is taking place in, not surprisingly, right? This MPF, mitosis promoting factor, peaks right before mitosis happens, right? So that's what kind of pushes the cell into mitosis. And then after mitosis, right, this protein goes away, right? You no longer can detect this protein inside of the cell all through the rest of the cell cycle until what? It's time to divide again. Then this MPF factor builds up again and pushes the cell into mitosis. Right? Now, obviously, this MPF factor is not going to be the only protein that's necessary to build up, right, in order to get into mitosis. What about all those other proteins that are actually needed to be able to carry out things like anaphase or telophase, metaphase, whatever, right? If you put all those on here, yeah, you'd see them going up, but it wouldn't be like they all go up in a big single peak. It'd be like all these different peaks, all sort of hinting at, yeah, them building up and being used during mitosis, right? But how is that coordinated? I mean, because that's a very confusing process. You're talking about, potentially thousands of different genes that have to be turned on at a particular time in sort of a synchronized fashion in order to get this intentional buildup or wave of proteins that are needed for the next phase. Right? So the key was discovered in the early 1980s. There is a group of very modest small proteins known as cyclins. Right? And so it is the rise and sudden fall of levels of these cyclins that actually serve as a synchronizing signal within the cell that controls or coordinates the expression of many hundreds, if not thousands, of other genes being expressed in synchrony. So the cyclin protein that's represented in blue here, you can see it rises, and as it rises, that's what stimulates the MPF protein expression to rise, as well as many other genes that are not shown on here, right? And likewise, when the cyclin levels drop, right, that's when the expression of the MPF also plummets, until in the background, the cyclin is building up again all through the cell cycle, and it gets to a certain point where it stimulates the MPF gene expression to happen again. So this secret is that there are these cyclin proteins, right? And instead of talking about hundreds or thousands of these cyclin proteins, you literally only need a handful of these to act as the master signals, right? To control different waves of other genes. So for example, during G1, when the cell is just hanging out, doing its normal differentiated cell functions, right? Hey, I'm a gland cell, right? My job is to make mucus, right? Right? So I don't care about replicating my genome. I don't care about mitosis. So the genes that encode for the proteins involved in DNA replication or mitosis, I'm not going to be expressing them, right? Because the ones I care about are the genes that I need to just survive. And my favorite gene is the mucin gene, because that's the gene that I express mucin proteins from, right? So I make a lot of mucus, right? Okay. Oh, but what happens if meanwhile I'm receiving signals from the outside, right? I don't want to keep saying I, like somehow I'm really the mucus gland cell, but the mucus gland cell, right, keeps receiving signals that are actually maybe encouraging it to go ahead and divide. So those signals might be little proteins that are being sent out by other surrounding tissues to encourage this target cell to consider going into cell division, right? So those might be like little growth factors. So epidermal growth factor, or EGF, is one of these critical little protein signals. So in the cartoon here, that's represented by the pink. So these little growth factors are sent out by other surrounding cells. And this cell here, the big orange one, is the one that's receiving it. How does it receive it? Well, on its or in its cell membrane, it has special proteins like this very elaborate looking weird thing here, right? As a protein that acts as a receptor. It is the right shape on this part of the protein to bind the EGF or the growth factor that's floating by. And of course, hey, we talked about this, when different proteins bind with each other, they kind of have to change their shape in order to accommodate the fact that now they're sort of pressed up against each other and all of their amino acid side chains have to get happy again, right? And so that causes sort of a ripple effect, an allosteric effect, where the bottom part of this receptor protein that's in the cytoplasm, it changes shape also. And suddenly now, right, other proteins that were kind of just floating by in the cytoplasm that were not the right shape to bind to this cytoplasmic part of the receptor, now the receptor has changed its shape and it is the right shape to go ahead and bind these proteins that are floating by. Right? So that's what these blue things are over here. So these things begin to sort of set off a series of, it's almost like communication, right? One protein binds, therefore it now can bind another protein. Ultimately, a series of proteins activate this RAS protein, this green one here, which has been hanging out on the underside of the cell membrane, right? So this is a membrane-associated protein because the RAS protein has hydrophobic sort of lipid attachments to it, right, that keep it kind of happily pressed up against the cell membrane, right? So this growth factor signal arrives, it changes the shape of this receptor protein, which then communicates to other proteins that sort of functionally link this to the activation of RAS. RAS changes its shape and it binds to GTP. So this is guanosine triphosphate. We looked at, ATP, adenosine triphosphate, and as an important energy molecule. This is guanosine triphosphate, right? So same thing, except for an adenine, you have a guanine attached to it, right? So that activates RAS and it goes ahead and kicks off a whole stream of other proteins in the cytoplasm that begin to communicate with each other. Ultimately, eventually talking to transcription factor proteins that then go and bind to regulatory regions like the promoters or enhancers of the genes that code for cyclins, right? So as a result of growth factor binding, you get this signaling that happens through a whole chain of proteins, eventually leading to gene regulation of the cyclin genes. So then you assemble RNA Pol II and you begin to express cyclins, right? And so the cyclin levels begin to build up inside of the cell, right? And when they hit a certain amount, they will then trigger the expression of a whole bunch of other genes that are kind of functionally linked to that rising level of cyclins. Let's look at and see how this works. Because to me, this is one of the most fascinating aspects of cell biology and how the cell cycle works, right? It is this idea of how does a single cell that has no brain, doesn't know anything, cannot really make decisions by thinking about anything. It can only respond to other molecules, right? Stimulating, activating certain pathways, inhibiting other pathways, right? How can this complex process of the cell beginning to replicate the genome, doing it perfectly, and then producing proteins that can then send it into mitosis, right? How can this all happen based upon biological molecules bumping into each other, right? So here on the left, you see that same cartoon that we showed before of cyclins, but now I'm showing you the handful of cyclins that are the critical ones. So you can see during, so the purple one, cyclin D2, right, rises up during G1, and it's the eventual rise and critical level of cyclin D2 that helps bring up all the levels of proteins that the cell is going to need for S phase. And that is what's going to plunge you over the restriction point, and the cell then is committed to going into S phase, right? Other critical ones are things like cyclin B. Cyclin B can be thought of. This is that cyclin that drives up the eventual production of mitosis promoting factor, right? So it starts building up and then its levels come plummeting down, right, in mitosis. So the other ones that are important are cyclin A, cyclin E, and cyclin D1. Right, so not too many to remember. I want you to be familiar with, you don't have to draw this, okay, but I want you to be familiar with how these different cyclins are sort of taking turns here, going up and down, right, as part of synchronizing their own group or subset of other genes that need to be now expressed so that the cell has proteins building up that are needed for the next stage. So how do these cyclins actually do this? The cyclins themselves, like I said, are very modest small proteins, right? They are not even enzymes, right? They don't even have any catalytic function, right? They're just these small proteins that bind to other proteins. The other proteins that they bind to are known as the cyclin-dependent kinases, or CDKs. Right? So kinase, we've heard that word before, right? The ACE part of it tells you, oh, this is an enzyme, right? And specifically, kinase means it's an enzyme that transfers phosphate groups to some other protein target, right? So these are kinases that will transfer phosphate groups, phosphorylating other proteins, but they only do this dependent upon cyclin proteins binding to them. And this is where the elegant key comes in here. This is amazing, right? Because once you learn this, you realize how wonderfully simple and elegant this is, and yet how effective it is. Let's say you have this brown blob here, which is one of these CDKs or kinases, right? So it's very happy transferring phosphate groups to some target, but the point is, it doesn't know what target to transfer the phosphate group to. It doesn't know what protein targets should be the target of its kinase activity because it's not the right shape yet, okay? But by binding to different cyclin partners, for example, let's start with the CDK over here in G1, right, where it's not bound to anything, so it's not doing anything. But if you have levels of cyclins building up, for example, the G1 S-phase cyclin, in this cartoon, it's representing that modest cyclin as red, but it would be essentially, let's just say it's the purple D2 cyclin over here, right? As it starts building up in the cell, because again, the cell is receiving all these growth messages that are causing the cyclin D2 gene to be expressing more and more of the cyclin D2 protein, right? As that builds up, what do you have? You have more and more of this cyclin D2 protein binding to the CDK. And that will then change the shape of the CDK. And suddenly now the CDK becomes the right shape to bind to a set of other proteins that before now, they were just floating around bumping into each other, right? But the CDK bound to this particular cyclin changes the shape of the CDK. So now that CDK is the right shape to bind to these other proteins. And what is it going to do? It's going to transfer phosphate groups, right? And phosphorylate those other proteins, therefore activating those other proteins, and those proteins will activate other proteins downstream of them, right? So that one critical phosphorylation event at the top has a trickle effect, and you suddenly have many pathways of proteins all being coordinated, right? Now look what happens. So those proteins that are eventually activated, those are the ones that will start turning on other genes. For example, genes that code for the proteins that are going to be needed in S phase. So things like the DNA polymerases, the different types of enzymes that are involved in doing DNA replication and making Okazaki fragments, things like that, right? So once you get into S phase, though, you lose levels of that red cyclin over here, right? So we'll pretend it's cyclin D2, it comes plummeting down. So as a result, the CDKs are freed up again, right? But what's happening in the background? Other cyclins are beginning to come up, right? So for example, I don't know, maybe cyclin A, right? You can see here begins to build up. And so that would be the green one in this case here, right? It now binds to the cyclin, to the CDK. So the CDK changes its shape again, but this time the shape is different, right? Because it's a different shaped cyclin that bound to it. So now suddenly the downstream protein targets that the CDK preferentially phosphorylate are a different set of proteins than what happened back in G1, right? And so the next wave of proteins get activated. Those proteins then turn on a different set of genes, and those might be the genes to express proteins that are necessary, right, to get out of S phase into G2. For example, these might be proteins that do things like detect whether the genome was properly copied during S phase. Many of these proteins might be proteins that can repair damage to DNA or mistakes in the DNA replication, right? So on and so forth, right? So here's another way of just showing the same thing, right? The CDK by itself, right? It has kinase activity, but because it's not the right shape, it doesn't bind to anything in order to carry out its kinase activity. But if you have this particular, so for example, this purple target protein up until now will just float by and sort of bump off of the CDK, right? Because they don't bind together. But if you have a cyclin that attaches to the CDK, together they become the right shape to bind to this target protein. And now the kinase activity of the CDK, right, adds phosphate groups. So it turns the purple blob into purple fortune cookie or something, right? So with the phosphate groups now this is the activated form that eventually now can go turn on other genes, right? Okay. So before I move on, right? So my point here, right, is hopefully if you think about this enough, right, spend some time thinking about what's going on here, you realize what a wonderfully powerful and yet surprisingly elegant and simple, right, system this is. You have these critical cyclin genes that are making these cyclin proteins that are ramping up in level and then crashing down at certain parts of the cell cycle, right? And by having enough of these proteins, cyclins, a particular cyclin floating around, right, you start binding to these CDK proteins that were already there, but just not doing anything because they're not the right shape, right? And depending upon which combination of cyclin and CDK proteins you get, then that CDK protein can do its kinase functions and target specific subsets of proteins, right? Triggering a whole series of pathways that result in coordinated expression of large programs or sets of genes to make proteins that are needed for the next stage of the cell cycle, right? All right, let's finish off today talking about apoptosis. So apoptosis is what we call programmed cell death, right? So of course, cells, if they get really stressed out, like if there's not enough nutrients, not enough oxygen, or there's exposure to toxic types of molecules that end up messing up the cell function, you know, those cells might die, right? Just because they can't function properly, right? But apoptosis is different. Apoptosis is when you have a cell that is still in its normal environment, has all the resources, all the nutrients it needs, right? But for some reason, something has gone really wrong with this cell, right? For example, right, if its genome gets messed up and you start having defects in your genetic sequence, it's really dangerous if that cell continues to live and make these weird mutant forms of proteins, right? Because maybe some of these mutant forms of proteins might do something like cause the cell to start dividing out of control, resulting in a tumor, right? So when cells detect that something has gone really wrong with their genome and they cannot fix it by the use of different, DNA repair types of proteins or enzymes, then maybe the cell needs to do the right thing and just kind of take itself out of the picture, right? And so to do so, our cells have evolved a program, a self-destruct program, right? It's kind of like in, science fiction movies. I like science fiction movies, right? When they have to abandon like the space station or the ship, and they have to do the self-destruct thing, right? They flip open a little thing and hit a button, and then and then the ship self-destructs, right? Usually in the movies, there's some sort of like robotic voice that says you have 60 seconds to get to somewhere safe, right? So then they run. So on the bridge of the ship, they hit the button, and then they all run to the escape pod or whatever. But of course, since it's a movie, something prevents them from getting to the escape pod, so it's all exciting, right? I don't know why they have to have the self-destruct button on the bridge of the ship. I don't know why they don't just have it like as a remote control in the escape pod, because that would make more sense, right? But whatever. But the point is, right, once you push that button, it kicks off a very specific program of events that leads to the destruction of the spaceship, right? But we're talking about cells here, When cells trigger the apoptosis program, it is a special series of events that lead to the rapid self-destruction of the cell. Okay? So this could be old or damaged cells that need to be, just eliminated. It can also be healthy cells during the physiological development of our tissues. For example, when we're in an embryo. Remember, we looked at a slide that sort of reminded us that in early embryonic development, we have things like a tail and gills, right? Like fish do, right? What happens to those tissues, right? How come you're not born with a long tail, right? It's because during normal fetal development, those cells that make up your tail or make up the gills, right, they undergo apoptosis as part of normal, healthy tissue development. Okay, so apoptosis is not necessarily bad, right? So why do these cells undergo apoptosis, right? Why is it this carefully orchestrated thing? It's because a damaged cell that just randomly blows up and bursts, that's not a good way of getting rid of it, because there might be weird things inside of the cell, right, that would just become released and affect cells that are surrounding it, right? Therefore, apoptosis is a carefully managed series of events to very, very carefully take care of destroying the cell. It's kind of like, when it's time to destroy a big building that has been condemned and they need to knock it down, right? You don't just go there and bulldozers don't just go and push it over, because maybe it'll damage nearby buildings or houses, right? So you have to get a demolition crew that knows how to properly set charges inside of the building, right? They know what they're doing, hopefully, so that when they do hit the button to blow the building up, it blows up in a very careful way and it causes the building to implode on itself, right? Rather than fall over and hit another building. So apoptosis is like that as well. So here are the four main steps, three main steps of apoptosis, starting from a healthy cell. As it begins to undergo apoptosis, the chromosomes condense, so it kind of collects all of its DNA into a nice tight sort of location so that the DNA can then be destroyed easily. The membranes begin to break down, so many of the proteins that are part of the membranes begin to be destroyed, and so the membrane begins to kind of form these funny little blebs, right, little side effects, little bubbles, right, of membrane. And then eventually the remnants of the dead cell, after all of the biological molecules inside are kind of chewed up and destroyed, you're left with debris, right? So even like a building implosion, a demo, there's debris to clean up afterwards, right? It doesn't magically, all the concrete doesn't just magically disappear. So once a cell finishes apoptosis, we have cells in our bodies like our macrophages that are constantly patrolling around, cleaning things up, right? So the macrophage will clean up all the little remnants or the apoptotic bodies. What is amazing is the entire process of apoptosis is very fast. It takes less than an hour from the time that the cell pushes that self-destruct button. Okay, so last slide for today, right? So apoptosis can happen from within the cell, meaning the cell itself detects that there are problems that it can't deal with, so it decides, all right, it pushes the button and it undergoes apoptosis. Or it could be that you have a cell that really is damaged and it doesn't know what it's doing and it's not actually triggering its own apoptosis anymore. Then you have to have other cells like lymphocytes. The T cells from our immune system come and detect that there's something wrong with the cell, and it tells that cell, hey, no, right? You shouldn't keep going. You need to undergo apoptosis. So that means the apoptotic signal can come intrinsically from within inside the cell that is affected, or it can come extrinsically from other cells telling the cell, nah, there is something wrong with you, right? You need to get rid of yourself. That's what this cartoon shows here. So here's the button, right? Apoptosis is activated, right? So extrinsically, right, you can have something like a T cell come along and present certain types of messages in the form of these death signals that are received by the death receptor on the outside of the target cell, right? Really cheerful, morbid name here, right? But it's appropriate because if this death receptor is activated, right, it activates a series of enzymes known as caspases. So these caspases are enzymes that digest or break up other proteins and other cellular components. So then these caspases will be activated. There are certain caspases that start the process, right? And other caspases that actually carry out the process. So initiator caspases activate, and they activate and turn on executioner caspases. And these executioner caspases, look, this one has a little mouth on it, right? These are the ones that are actually going to destroy different components of the cell, right? So that happens from an extrinsic signal coming in. Inside, right, if the cell is detecting its own damage, for example, there's DNA damage down here, there are certain proteins that will detect that the DNA is damaged, cannot be repaired, right? And those proteins will activate the release of cytochrome C from the mitochondria. You're like, cytochrome C, mitochondria? We just finished studying that when we talked about mitochondrial aerobic respiration, right? And you're like, cytochrome C, what is that? Remember, that's the little one of the electron transport chain components that carries electrons from one of the big brown electron transport complexes, big brown blobs, to another big brown blob, right? So importantly, cytochrome C, the only place it normally should be inside the cell is inside the mitochondria, right? But if proteins detect that there's too much DNA damage and the cell needs to undergo apoptosis, the mitochondria releases cytochrome C into the cytoplasm, right? And so this is now the sign that apoptosis is about to happen. This cytochrome C binds to other proteins, these adapter proteins, apaf1, and they form this kind of wheel that we call the apoptosome, right? It's often called the spinning wheel of death, right? I'm not making this stuff up, right? That's what they call it. Because the spinning wheel of death then will go around and activate executioner caspases, right? That normally are folded up in an inactive form. So these caspases often have extra amino acid sequences as part of their peptide chain that keep their mouth or the catalytic site covered up, right? So we call them procaspases. But this apoptosome, the spinning wheel, will come, right, and bind to this, remove those extra parts of the peptide, and now the mouth part of the caspase is exposed. So it goes from being a procaspase to an activated executioner caspase, right? And then that's what carries out apoptosis. Okay. All right, let's go ahead and stop there today.